All right, so good morning everybody, and welcome to this GYAN lecture series. We're going to have lecture 6 on combining dual receptors for enhanced sensitivity. First of all, brave effort all of you making it to the auditorium despite the inclement weather that you have outside. You might find my voice a little bit cracking today. It's just because I've got a bit of a sore throat, not still used to AC and fan running at the same time. But anyway, I hope I'm able to explain what I would like to explain as part of this dual receptor strategy that I want to present today. So as I promised yesterday, this is going to be a very interesting lecture. I'm also kind of giving you some really some very fresh reports, some fresh results in this. Some of this material that I'm presenting today is basically unpublished work as well. So fresh from the press, as they say. I'm going to be presenting this whole lecture series to you, this lecture to you. In terms of course structure, I mean this all has been changed quite a lot. So I think you are probably well updated through the group that we have. So please follow the group. Don't worry about this course structure. We have to move things around to make sure that you have a very pleasant learning experience. In terms of suggested reading, there are 2 articles I would like to I would request Khalid to share with you. Both of them should be on the Dropbox folder, and these are basically 2 papers. One of them is on the SARS-CoV-2 virus label-free electrochemical nanohybrid MIP apta sensor, and the other one is one which has been published from my research group on Alzheimer's diagnosis beyond cerebrospinal fluid, which is basically a paper telling you how we could move away from painful lumbar punctures and start doing point-of-care diagnostics in simple samples such as the blood samples. All right. Again, this is using MXene and a redox system and molecular imprinted polymers. The main objectives of this particular lecture is to explain to you the rationale behind the use of dual receptors. You know, we've been talking about biosensing devices. We've been talking about the fact that at the heart of the biosensing devices are your bioreceptors, and the thing that you get with those bioreceptors is you get the desired, I mean you would like them to be very selective, you want them to give you the desired specificity, but then you can play around with when it comes to the sensitivity. And sensitivity is something that's a very important performance metrics, and you need to focus on that in case you would like to, and this will vary from the application to application. So in the case of glucose, we saw that the concentration are normally in the order of millimoles, which is 10 to the power minus 3 moles. When you look at biomarkers, which are normally seen in blood or in the cerebrospinal fluid or the skin interstitial fluid, the concentrations might be very, very low. So the idea is to develop biosensors which can give you the desired dynamic range, which can give you the desired sensitivity as well, where the limit of detection of your biosensors should be able to detect the range which you are normally focusing on. For example, in glucose, you're trying to develop biosensors which can give you a dynamic range of 0 to 20 millimolars, because that's the concentration of glucose, and that's the physiological concentration that you normally you normally come across in the human body. But the biomarkers, they are definitely not going to be very high in concentration. They would be something as low as picograms per milliliter concentrations. So you need to adjust, you need to monitor, you need to develop your biosensing device, and there, as I said, the core there is the receptor, and then how do we change the receptor, how do we make it much more stronger to kind of cover and make it more sensitive, make it ultra sensitive. That's what we're going to understand from this particular lecture here. We're going to be looking at the integration of dual recognition systems, basically dual bioreceptors, and I'm going to give you 3 good examples. MXene, PDA, MIP sensors, the one that's the paper that I've already put on the, I showed you before. Some work on highly porous gold and polyphenol red and polypyridine based MIP sensors, and most important, as I said, red hot from the press is going to be some results freshly produced in the last week by my research group on aptamer, combining them with the nano MIPs, and again having the complex system there. And then we're going to look at the discussion, and we're going to discuss on the implications. Hopefully you will understand how using dual receptors allows you to increase the sensitivity, selectivity, and how this will impact the translational potential of your product. Okay, so first of all, we ask ourselves the question, why do we need dual receptors, or why can't we manage just with one receptor? Well, sometimes when you have single bioreceptors such as antibodies, aptamers, or just MIPs alone, you have limitations. You can have limitations in terms of stability. Your bioreceptor may not be stable enough The other problem you have, the other limitation you have is the selectivity. You're not getting the desired selectivity. And the third problem is the signal to noise ratio. You may not be getting the required signal to noise ratio. You may not be getting ultra sensitive detection for the dynamic range that you're aiming towards. So you then employ dual systems, because then they allow in improving the binding affinity, right? The stronger they bind to your target analyte, to your biomarker of interest, the better results you will get. And it also reduces cross reactivity, and it enhances signal amplification. So that's why we employ dual receptor systems. Now we are all scientists, right? We would like to see some evidence here. So first of all, let's have a look at what are the possible combinations that you can have for the dual receptor system. The possible combinations you can have a natural antibody and you can combine them with an aptamer. You will come across different terms these days being employed: nanobodies, aptasensors, aptabodies, and stuff like that. When you combine an antibody with an aptamer, it becomes an aptabody. Similarly, you can combine a MIP with an aptamer. You can combine a MIP with a redox active polymer. Again, this is a question mark here. And then you can also have nanomaterial systems such as MXenes, which are two-dimensional structures, two-dimensional systems. You can use those scaffolds and then combine them with MIPs or enzymes. And I'm going to be giving you some examples there. So again, coming back to this question, why do we need dual receptor systems? We are all scientists. We all believe in evidence. We don't believe in things happening by chance. So here's the evidence on your screen. Apologies for the small font here. This is an example. This particular column is that on dual MIP aptamer sensor systems, wherein you combine two bioreceptors together. This is your target analyte. This is your detection mode. And then on the right hand side, you have limit of detection when you just have the single bioreceptor. And then what happens when you combine the two bioreceptors, and the DOIs, the slides will be shared with you. The DOIs are where you can pull the papers out and you can read them individually, depending on if the biomarker is of interest for you or not. All right. So let's have a look what happens when you combine aptamers with MIP hybrid. This is again looking at the PSA. Anybody here knows PSA? It's prostate specific antigen. This is a cancer biomarker for prostate cancer, right? Very, very important. It's normally measured electrochemically using electrical impedance spectroscopy. Now, when you have a single bioreceptor, your limit of detection is around 3 picograms per milliliter when you just use the aptamer. However, when you combine the aptamer with a MIP system and make it into a dual bioreceptor system, your limit of detection goes down to 1 picogram per milliliter. Now, these things make a huge difference when you have clinical samples. Your median value could be somewhere around 1.5, 1.8, right? So going down from 3 picograms per milliliter to 1 picogram per milliliter is definitely very useful. If you want to have a look at this paper, you have the DOI on the screen here on the right hand side. Let's have a look at the second evidence that we have here. What happens when you combine aptamer with a MIP on cuprous oxide nanocubes, which is basically a nanoparticle system. And this is for a particular mycotoxin, which is your aflatoxin B1. Again, you're using electrochemical impedance as the detection mode here. What you see here is from 0.9 picograms per milliliter using the aptamer alone, you're able to push down the limit of detection to 0.012, roughly about, I would say roughly about 8 times better sensitivity here, right? So what's happening here? Combining the aptamer with the MIP is giving you an enhanced limit of is giving you an enhanced sensitivity and giving you a lower limit of detection. You're able to push it down, and again the DOI for this is mentioned on the right hand side here. The third one here is an example where an aptamer has been combined again with another MIP nanohybrid system. It's mainly used for SARS-CoV-2, and this is one of the papers that I've given you as suggested readings, because what you can see here, the technique is the same, electrical impedance spectroscopy. You are able to look at 9 into 10 to the power of 3, which is roughly about 9,000 PFU per milliliter, which is just with the aptamer. From here, from 9,000 you're going down to 3.3. Can you imagine the amount of, how much, by how many orders? Nearly 3 orders, nearly 1,000 times, or nearly 3,000 times increase in sensitivity. This is a fantastic piece of paper, fantastic piece of work that has been published. Again, I think it's Microchemica Acta, which is the journal in which this has been published. Have a look at this paper as well. Another example for dopamine, alone with the aptamer, you're getting 63 nanomoles, but as soon as you use a MIP system there, again with a nanoparticle system, you have 47 nanomoles sensitivity there, and again this paper is reported here. And the last example you have is that of insulin. Insulin, as we all know, is a peptide hormone. Alone with the aptamer, you're getting a sensitivity of, or you're getting a limit of detection of 1 nanogram per milliliter, and when you use this with a MIP and a magnetic nanoprobe, you're able to get this, able to get a limit of detection of 0.5 nanograms per milliliter. You've almost doubled it, right? So this is, you're making your systems much more sensitive. So these are the evidences which encouraged us as a research group to look forward into dual receptors, and I'm talking about the project that we have, which is the UK Japan MRC AMED project on early diagnosis of Alzheimer's. Again, there the concentration levels we are looking at are picograms per milliliter kind of concentrations, which is picograms is 10 to the power minus 12, right? Just to put things in the right order. Nanograms is 10 to the power minus 9. So I hope the scales are, because I do appreciate that we have a wide audience, we have people from different backgrounds. So just to kind of just to make sure that everybody understands the units that we are going to be using here. Okay, let's have a look at case study 1. This is again from my research group, which is on amexine MIP PDA sensor. Basically, we've used amexine here with polydopamine, and the target here is small molecule analytes. In this particular case, we are doing tau 441 protein, which is a protein with 441 amino acids. The nice thing with this particular system is you have a high surface density of the amexine, you have the PDA, which is polydopamine, which provides the functional groups so that the MIPS can anchor to that, and you have an improved sensitivity. That's the overall summary. Let's have a look at how this has been done in the group, and this has been published as well, so you can please feel free to read this paper. It's a part of your suggested readings. Your amexine based redox systems and molecular imprinted polymers. So we started off by just using, in this particular case I'm giving you an example wherein you have you're just working with one single bioreceptor, which is going to be MIP in this particular case. There's nothing else here. How do we do the MIP formation? You have your screen printed electrode. The screen printed electrode has been cleaned and rinsed with water, and on that you normally electropolymerize polyaniline, and this polyaniline is polymerized in the presence of your protein, which is your tau 441 protein here, and then you bring in the oxalic acid and you remove, as I think I've already explained this, how the molecular imprinted polymers work, and you remove that with oxalic acid, you remove the template, and then you finally have a templated molecular imprinted polymer to which you would expect your 441 molecules, which are your target biomarkers, to come and bind to that, right? That's happening here. Results look good, and as you can see here, we're looking at femtograms per milliliter concentration here, right? I think it's not very clear here. I think I should be clear in the next slide here. Yeah. So if you look here, yes. So if you look at this concentration here, you're looking at femtograms per milliliter concentrations that you're normally getting here. And this is normally done, these studies were done in PBS, right? It's normal, this is what everybody does, and in artificial ISF as well. The idea of doing it in artificial ISF is these are in vitro studies, so that you can use these as wearable sensors as well, where you're going to be monitoring in the skin interstitial fluid. But that's if you have the evidence that your biomarker of interest are actually changing over time with and they're changing rapidly, so that they really require continuous monitoring as well. Okay, so I mentioned, and then in the next step what we did was we did this just alone with the MIPS, with the molecular imprinted polymer as the bioreceptor, and then we combined our molecular imprinted bioreceptors or molecular imprinted polymers with the amexines. Now these amexines are basically two-dimensional materials, right? They are very layered kind of structures there. They are nanomaterial systems, and they basically consist of a transition metal, and they normally have carbides of transition metals normally in these there. And these amexines have been seen in many different applications. They've been used for sensors, they've been used for biomedicine, electromagnetic interface shielding, they've been used for electronics as well, your 5G antennas, your electronics and your smart textiles. They're used for energy storage, they're used for environmental applications, and also for optoelectronic applications as well. So nice, this is one of those two-dimensional materials that my postdoc had already some experience of working with. He knew how to handle it, so he decided to introduce a nanomaterial system to add it to the existing molecular imprinted polymers that we had made out of polyaniline here. So these are some results here. As you can see here, you have the screen printed gold electrodes. You take the screen printed gold electrodes, and then which has got your PDA, and you've also got your molecular imprinted polymers, and then you actually assemble that on top of your vanadium based amexine system that you have. And as a result of that, we see a great enhancement in the sensitivity. We're looking at log concentrations here, so we are able to increase the sensitivity, and we are able to do much more better. We are getting much more better linearity. If you look here, you're going from nanograms per milliliter to femtograms per milliliter. So what you've done is you've increased the dynamic range here, right? So earlier you were only getting you're only able to detect in the lower region. You're only able to detect in the femtogram per milliliter region. Now here, by combining the two bioreceptors, or in fact combining one bioreceptor with a nanomaterial system, you can show that you've increased the dynamic range. So you can use this particular system not just for early diagnosis, but you can also use it for prognosis, right? People who already are suffering from the disease are undergoing some form of a chemical intervention, some form of a therapeutic intervention. Now, how is the disease progressing, right? How do you find out that? Because obviously people who are suffering from Alzheimer's disease will have a much higher concentration of the biomarker, right? So if you just are able to do early diagnosis, that's fine, but what you would also like to do is also do a prognosis of what's going on. How is the disease progressing? Do you have sensors which can tell how is the body reacting in response to the drug therapy that's going on there, right? And these days we all know there are a lot of drugs that have been introduced for treating and managing Alzheimer's disease as well, which can slow down your neural degradation. But again, as I said, you need the evidence, and that evidence can only come through prognosis, through prognostic screening, right? You keep on monitoring the levels and see, okay, the person is on drug, is he reacting to the drug or not? Should we increase the drug or not? So you need not just early diagnosis, you also need prognosis. You also need to be able to follow the disease through the biosensing or through the diagnostic devices that you normally have. So that is the case study, or that is the example of the first one, which is the MXene based redox system and molecular imprinted polymer. Now we move to the next one, which is basically an example of highly porous gold with polyphenol red and polypyridine based MIP sensors. The idea again here is the same. MXenes have got a problem. MXenes have got transition metals, right? When you have something which is transition metals, there's always a bit of reluctance there that even if that nanomaterial system isn't going inside your body, you still have some reluctance even if it's coming in contact with capillary blood, right? So what do you do? And the other problem is MXene doesn't have very good anti-fouling properties. Just even when you drop even when you put a drop of blood on an electrode, you will see some form of biofouling, which is basically the proteins in the blood would go and adsorb on the surface of the sensor. So you should be able to do measurements without any interference from the proteins in your blood samples. So what we did was, after we saw that MXenes was, we did our first work when we were at Swansea University on the use of MXenes, and then when we moved to University of Liverpool, we decided to change the design, the architecture of the sensor, and we said let's make something which is ultra sensitive, which gives you the desired dynamic range for monitoring of Alzheimer's disease, but based on something which is more biocompatible. And that's when we came across this particular material, which is called as highly porous gold, right? HPG. And we combine this with a MIP system, which is basically a polyphenol red based MIP system, and which has also got some polypyrrole also in it. And as I said, as you can see from the image, the idea is that you have a person who doesn't have to go to a specialized hospital, right? If you're talking about Alzheimer's, right, the gold standard for Alzheimer's, as I might have already mentioned, is basically wherein a patient goes into a specialized hospital, you get the highest concentration of the biomarkers of Alzheimer's in, louder? CSF, cerebrospinal fluid. How do you extract the cerebrospinal fluid? Through lumbar puncture, right? And lumbar puncture is it's a very invasive method, right? So until we have pioneers working in the area of sampling methods, give us something easier to get the cerebrospinal fluid, it's going to be a very painful method, and you need a specialized person for that, because you basically have a thick needle pushed into your through your vertebra into your spinal cord to extract the cerebrospinal fluid there. Right? Not a very pleasant experience. You can damage a nerve, right, in the whole process of trying to access the fluid and stuff like that. So therefore what we want to do is move away from cerebrospinal fluid into something that's easily accessible, right? What is easily accessible? Okay, I know the first answer would be skin interstitial fluid or sweat, right? But sweat doesn't have the biomarker. So what is our next best available sample matrix? Then blood. Which blood? Venous blood, is that more easily available? Capillary blood, because people are used to pricking their fingers to draw capillary blood. So if we can do measurements in capillary blood, or for that matter, even if you went to a GP practice, if you went to see your physician there, and if he can easily draw some venous blood, it's not rocket science. I mean, he just needs to puncture your vein and just draw a little bit of blood there. I mean, I know they normally they have this practice of doing 5 milliliters so that they can do all kinds of tests on you, but you know, you need a very small amount of blood there. This is a very simple. This cartoon basically is showing a very simple case thing wherein you are able to do all these measurements in capillary blood, right? And we are actually very interested in capillary blood as well. Okay, so this is basically a representation of how the work was done. We are basically taking capillary blood and we are using our what we call as the PCB electrodes that you see here, which has got two different working electrodes, one counter electrode and one reference electrode, and we basically these has been functionalized with your nanoparticle system and your MIPS on top of it, and then we are using all the data that comes in there. We do a lot of clinical studies, and then we use a machine learning regressor tree, and these are things that you'll have to speak to my PhD student about, because this is all his business. And then from that data analytics, we try to predict actual values as well. So you need to train your algorithms, machine learning, and get your AI tools there, which then help in predicting. The reason we also need to bring in machine learning and AI, I would just spend a few moments to explain that as well. Now what happens is you have the biomarker levels, right? So you can say the biomarker level is at the median value is around 10. But in a healthy person who's kind of hitting the age of 80, the levels could be 100, 106. So you need to do a correlation with the age as well. Therefore, you need to bring in the different tools which can, if you know that there is a trend there that the biomarker levels will increase with age, and it could mean that the person has got high biomarker levels, but the person is he's a control person. He doesn't have AD there, and that can be certain through cognitive tests, right? I'm talking about biosensing devices here, but remember one thing: in clinical situations, if we have a clinician amongst us, that clinician will say you that it's not based on one parameter. You look at multiple parameters. You look at cognitive tests. You look at different biomarkers. And it's not. That's why we talk about multi-analyte sensing. Very, very important. Multi-analyte biosensing. You look at multiple analytes. If you have the amyloid proteins, you don't look at just amyloid beta A beta 40. You look at the ratio of A beta 40 and A beta 42. One of that will be overexpressed. One of that will be underexpressed. Right? You look at that particular ratio, you get a number for that, you look at the value of the NFL, you look at the value of the p-tau proteins, so you have multiple analytes, and then you look at the cognitive tests as well. And then you make a decision, how is the disease, number one, so you have to do patient stratification. You need to ascertain, like in cancer, you have different stages, right, based on the severity of the cancer. Similarly, in all the diseases, you have a certain parameter, that you have a certain standardization of how you classify them according to the severity, right? So you need all the tools that you can get. You need multi-analyte biosensors so that you can get all the information you need to ascertain how severe the case of Alzheimer's disease is, right? And therefore, you not only need your biosensing devices, you also need your machine learning tools, you need your AI, so that you can build what we call as a clinical decision support system, a CDSS, right? That's very, very important. Okay, so let's have a look at what happened. What did we do in this particular case here? So here again, the system has got highly porous gold. We're using polyphenol red and polypyrrole and an MIP. Now, one of the things that I forgot to mention before when we did the first case study is, when we had this particular system here, what we also had to do is, to be able to read, we had to do electrical impedance spectroscopy and differential pulse voltammetry, but we had to bring in a redox probe. We would bring in a solution of Fe2, Fe3 plus, so that we can basically look at the impedance measurements there, right? So we needed an external redox probe to do the readings, to look at the surface. So as I said, what is happening is you have the surface. So what you have is you have the metal surface. On the metal surface, you have your nanoparticle system. On the nanoparticle system, you have your polymer. This molecular imprinted polymer has got your cavities, right? And just to kind of, so when you measure the impedance here, you got cavities coming directly in contact with your nanoparticle system, coming in contact with your metal surface, right? So if the electron transfer is going to be very easy. You have a solution, so you bring in a solution of Fe2 plus, so you can easily see the oxidation and reduction. There's very low impedance there. But the signal starts to drop as you have binding of molecules onto the sites. So this now has now increased the impedance. And as you keep on binding more and more molecules to the sites, you then, what you're basically doing is you're increasing the resistance, you're increasing the impedance there, right? And that's normally measured in the form of a signal drop, right? So you see this signal loss thing that we see here, that's basically how the signal loss is seen. And the nice thing is this signal loss nicely correlates to the concentration here. So in our first work, we were using an external redox probe on top of the whole system that we had, and we wanted to move away from that. So we used polyphenol red and polypyrrole, which are themselves redox polymers. So now we don't need an external redox probe, if that makes sense. Okay, so we've done this, and what we have is, as I said, these are redox active composite polymers which have got a very high surface area. For you have getting a very high surface area for highly porous gold. The other thing with highly porous gold is it's got very good anti-fouling properties, right? Nothing sticks to it that much. And then you're basically using machine learning models for classification and quantification, and in terms of performance you're able to measure as low as 498 or 0.5 500 femtograms per milliliter limit of detection. And there's also this something called as ROC AUC, area under the curve, and ROC is receiver operating control. So normally the good value is anything above 0.8 is considered to be a good value here. Okay, so that was the summary. This is, what is this? Anybody? What does it look like? Highly porous gold. So these are SCM images of highly porous gold. Just to show you exactly, when you zoom in, right, you can see this. So this is the normal gold, and then this is how we have look at the highly porous gold. I think the scale bars are slightly different here, but these are the different types of. So this looks more like a planar gold. These are how we made different types of highly porous gold, and you can see that. So as I said, the advantage of the nanomaterial system is you get a high surface area to volume ratio. Does everybody here understand the meaning of high surface area to volume? I can give you another good example which you can easily relate to. Can anybody tell me that particular part in the body which, if you keep on stretching it, can take you all the way to the moon? DNA. Fine, that's fine. And I said part of the body. DNA is more kind of a molecule. You're talking about molecular level. Pardon? Anything else? Yeah, nerves, neurons, the same thing. Have you heard about microvilli in the intestine, in the small intestine? What are they? They are basically foldings, right? So if I go back and if I try to show, so if this is it's like a tube, right? If you don't have any inner lining, you see this will have a particular surface area. You can increase this surface area if you have the inner lining which has got finger-like projections, right? Why do we have this finger-like projections in this thing? To increase the absorption, right? That high surface area to volume ratio. Within the same volume, you have a much higher surface area, right? That's what happens with highly porous gold as well, right? Within the same planar gold, you have porosity there. You have pores, and you have these kind of granular structures, right? And on top of this, so this is the nanoparticle system. It enhances the sensitivity. It enhances the signal. It amplifies the signal. And on top of this, you have your molecular imprinted polymer. So you're getting the benefits of two different systems. You're getting the benefit of a highly sensitive bioreceptor, which is your molecular imprinted polymer, sitting on a nanoparticle or a nanomaterial system which is giving you the high surface area to volume ratio. So you're basically increasing the density of your binding sites, right? Rather than being plain, it's up and down. So what's happening again, remember the example of microvilli, right? It's up, you're basically having a much high surface area to volume ratio there. These things together allow you to get a much better system there. Okay, so this is your gold, and you can see this whole scale bar is 200 nanometers. So if you look at these particular granules, there's something ranging between 50 to 100 nanometers kind of scale that we are looking at. So we use this, and then this particular system was mainly designed for p-tau 181. When I say p-tau, anybody knows what does the P stand for here? P is phosphorylation. So which side is it phosphorylation? The biomarker tau 441 is basically a protein which has got 441 amino acids. This one, p-tau 181, is basically where the amino acid has been phosphorylated at site 181. That's what gives it the specificity here. All right. So you can see here we use the same system that I mentioned. You have your your planar gold. On top of that you have your HPG, highly porous gold, right? And then on top of that you have your molecular imprinted polymer made up of phenols, polyphenol red and pyrrole. Again, why do we need polyphenol and polypyrrole? I won't go too much into the details. The papers should be coming out soon. We've already communicated these papers. The papers will come out soon, so you'll get all the information that you need. I know that some of you, I don't want to go delve down too much into the basics, but as I said yesterday, you can tune the materials. It depends on what your target analyte is. Your target analyte could be something which is highly hydrophilic or something which is highly hydrophobic. Combining polyphenol red with polypyrrole gives it the required electrical conductivity. It also gives you the required mechanical stability as well. So sometimes, these are molecular imprinted polymers. You want them to maintain the shape, because here your binding depends on the shape, the geometry, the way the whole thing is three-dimensionally arranged, and it also depends on the charge centers there. Because if you remember yesterday when I gave you that lecture on molecular imprinted polymers, I mentioned about the importance of the interaction mechanisms. I mentioned about the forces that allow you polymerize around it. I mentioned about the hydrophobic interactions which will repel, electrostatic interactions which will attract, hydrogen bonding interactions which will, so you want to maintain that whole thing as if it's staying in a native state. So for that, the most important thing you need is your molecular imprinted polymer, the polymer that you're using to template has to have a certain level of stability. And in our case, we are using something which is redox active. So what you don't have to do is, you don't need to add an external redox probe. You can do all the measurements just by looking at the surface characteristics of your design, of your particular device there. You can use differential pulse voltammetry. I mentioned to you already that we have stopped using EIS. We only use EIS when we are investigating the whole formation, but otherwise we normally use differential pulse voltammetry or square wave voltammetry, because they are more gentle on the system. You're not applying an AC up to from the whole range of AC sinusoidal waves that you normally apply in electrical impedance spectroscopy. All right. So then here are the results in front of you. Very happy to show you this. This is basically your tau protein. You can see the one which is the one which is in the red. This is your human plasma. The one which is in the black, this is again human serum. So basically you can measure this, and we've also done some studies in whole blood as well. So if you just draw some blood and if you just transfer it straight to a biosensing device, no, it never happens. I'll tell you that for sure. You might have seen people take blood and then they transfer it into those ampoules, which then does the separation, which does has got those anti-clogging EDTA and stuff like that, and then you do the separation into plasma and serum and things like that. You can do a lot of those things. So normally you would find most of the p-tau distributes itself in plasma. You have a much higher concentration in the plasma, but we've also shown that we can also measure in serum as well. And the limits of, you can see here concentrations. What limits are we looking here? What is the sensitivity here? What's the dynamic range? We are looking from 0 all the way to 25 picograms, right? Is that the right range? How will we find out what is the right range? We look at the graph below, right? This is the violin plot for Alzheimer's disease, and this is a violin plot for non-Alzheimer's here, right? As I said, everybody has got a different way of, everybody's body is different. Everybody expresses biomarkers in a different way, right? This is the median value for a person with Alzheimer's, which is around between 4 and 6, which is around 5 picograms per milliliter. This is that for a non-AD or a control sample. This is around roughly about 3, and this is roughly around 5.5, right? This is 5.5 and this is around 3.0, right? Picograms per milliliter. Look at the difference, how subtle the difference is. Now I've given you violin plots here. What are these violin plots for? The violin plots normally say these are based on the studies that have been done on multiple participants, right? So what we've seen is people with Alzheimer's, the median value is, the values range from here all the way till here, right? You can have healthy controls, the values range from something around 1 all the way to about 6. So you can have those outliers as well, as I said, right? So, but here if 5.5 is the cutoff, this is the median value, and here you're getting a value of roughly 6. But again, this person is still a healthy volunteer, or a control, not an Alzheimer's person. Why are the levels high here? Because this is something related to the age, right? So you need to do the age related correlation as well. These violin plots show you the width of the violin. This width here is actually telling you that majority of the people, you know, how you get the median value, majority of the people have got this value of about 5.5, and this violin plot shows here the same thing in healthy volunteers is around 3. This is where the majority of the things sit here. This is how you read the violin plots. You will come across these violin plots. Nice way of describing your results, because here you are also factoring all the other, you know, you're not only telling about you know, the range, but you're also telling about the frequency of that range as well. Okay, more people with Alzheimer's disease have got a value of 5.5. That's the median value, right? Because what does a clinician want to know? I mean, if you send it to an analytical chemist, the first thing he'll ask you is, what is the cutoff? At what point do you differentiate between Alzheimer's and non-Alzheimer's, right? And then you say, well, there's nothing like that. There's no one particular value which will help you decide here. You need multiple. That's why I'm trying to stress here. You need multi-analyte sensing. You look at multiple biomarkers. You look at their levels. You literally have a spreadsheet, and then you also have the cognitive tests. And then the last column is age. And you say, oh, the levels are very high. The levels are around this one. In principle, this person should be AD positive, Alzheimer's disease positive, but he is not AD positive. Why? And then you go and look at the age. Oh, this is somebody who is 85 years of age. That's why he's got a high level of p-tau 181 protein in it. All right. We are now working on p-tau 217, which is now a WHO recommended biomarker for Alzheimer's, right? But again, as I said, just working on one biomarker does not help you, and that's why when you do your practicals, look at the design that we're going to give you. We're going to give you some PCB electrodes to work on. Look at those designs and ask the question, why do we have multiple working electrodes? We may not have the potentiostat right now to do all 4 electrodes, but at least what we can do is we can try and see if we can get some good values there. At least we can give you some experience of working with multi-analyte sensors or multi-electrode sensors, right? Okay, so this is the example that I wanted to give. Look at the limit of detection here. This is femtograms per milliliter, and this is picograms per milliliter. What's the difference between these two? Order of 3, right? 1000 times, right? So we are our limit of detection, so we can actually go down much more lower, and we can measure very low concentrations. Have we got the desired dynamic range? We would think so, right? But when we went to Japan and did some joint experiments, as I said, some of the values were around 108 picograms per milliliter. Simple. Can't do anything about that, right? So what do? Extend the dynamic range there. So we are now working on extending the dynamic range from 0 to 100, and anything about 108 we would say definitely positive for Alzheimer's disease, but please check the age to see the correlation there. All right. So that's the second example. Now I give you a third example, which I said is basically on neurofilament light chain, right? This is an example. I think we're okay for time, right? Basically, this is another biomarker for Alzheimer's disease, and I can tell you off the record that from our studies done in the UK, Japan. So what we are doing in the UK, Japan project is we are looking at the correlation of the biomarkers in different sample matrices, right? We are looking at capillary blood, we are looking at venous blood, and we are looking at skin interstitial fluid. All right. Now, in Japan, they have the technology to be able to express Alzheimer's disease, human form of Alzheimer's disease, in mice models. Why is it useful to study mice models? They have a short lifespan, right? You can start looking at the disease progression and then extrapolate it to humans, right? You can start looking at the disease progression in a very narrow period of like maybe 3 months, right? You do a study over 3 months. You have the mice model which has been expressed with the human form of Alzheimer's, and then you look at and you keep on measuring the venous blood samples. You keep taking venous blood samples. You keep taking blood samples from venous blood samples can be taken from rat tail. Capillary blood samples, you can take that from the mice as well, and then you can use microdialysis probes to extract out the interstitial fluid. I wouldn't use microneedles here because microneedles for extraction are not the best because the mice skin is very thin. I mean, I remember doing the microdialysis and I ended up going into the peritoneal cavity. It's so thin. So you need to stay away from that. So we've actually refined microdialysis, and microdialysis. We've now got a very good method of having the microdialysis wherein the rat is very much moving around and you can still sample from the skin interstitial fluid. And one of the nice things that has come out of the whole study, I mean, forget about the interstitial fluid, so I'm not talking about wearable sensors here. One of the nice things that has come out of the studies is that there is a very good correlation of the NFL in capillary blood and in venous blood. Is that useful information? Yes? How many of you say no? No, everybody agrees with me, that's very amusing. Why is it useful information? Louder please. Do you have a microphone? Because you can? Absolutely. So it makes your sample access much more easier. You can use a lancet and you can get capillary blood. If the same concentration of, let's say, if something is found in the skin interstitial fluid, right, why would you want to go and sample into the blood, right? Because skin interstitial fluid can be accessed in a minimally invasive. If something is found in the sweat, why would you want to go and access the skin interstitial fluid underneath the skin? If something is found in the skin interstitial fluid, which can be easily accessed using minimally invasive techniques, why would you want to start using needles and lancets and go into capillary blood? If something can be found in the capillary blood, why would you want to go and look into the venous blood? Especially if you know that there's a good correlation and you have sensors which are ultra sensitive enough, which are giving you the limit of detection, which are giving you the sensitivity, and you are able to cover the dynamic range, right? So NFL is one of those very good molecules, biomarkers, neurofilament light chain, one of those biomarkers which actually we've seen that in the animal studies that we've done has a good correlation in capillary blood and in venous blood as well. NFL is also a biomarker for Alzheimer's disease. So if you had a list of biomarkers, established list of biomarkers for which you have gold standards, so the gold standard of measuring these whole biomarkers is a technique called as SIMOA. Anybody heard about SIMOA? Yeah, it's single molecule analysis, right? And what does it? What exactly is it? It's basically a glorified ELISA system there, which is working on magnetic beads, right? So you have like something like a CD, and you have your magnetic beads. You trap your it's enzyme link. You got your enzyme and everything mobilized on it, and you spin it, and then each bead goes into a single reservoir, and then you're looking into single molecule. That's why it's called a single molecule analysis system. Okay, and again, it's an optical system. And do you know how much is one of this gold standard SIMOA kit costing? It's 200K. And when I say 200K, I'm talking about 200K pounds, right? And what we are trying to do as part of the UK-Japan project is to replace this with a 20-pound kit, which can be done with electrochemical sensing, right? Why? Because you can make your screen printed electrodes or your PCB electrodes for as less as 9 pence. You can have your potentiostats, which can be made for as less as 15 pounds, and then you just so the device price would be 20 pounds, and that will replace a SIMOA thing. But for that, we need to have biosensors which are ultra sensitive, which are reliable, which are reproducible, which are accurate, right? So it's a very ambitious target, but it's not impossible. It can be done. If I can't do it, one of you would take over and should be able to do it. Okay, all right. So coming back to case study 3, number 3, which is on neurofilament light chain. The dynamic range of this is 16.21 to 100 picograms per milliliter. This is what is stated in the literature, right? But the actual range is, I think, it's anything, and this is the unhealthy levels is over 30 picograms per level. So when the concentration goes above 30 picograms, then you say this is where it's 30 picograms per level. 11.5 is actually what we call as the median value for a healthy volunteer. But as I said, you may find samples from a healthy volunteer values of around 108 picograms per milliliter. That's why you use those median values, right? Because there's a huge distribution there. You can have 108 picograms per milliliter concentration as well in healthy volunteers, but who are at a certain age as well. Because neuronal degradation is something which happens in the body, right? Keeps on happening, right? And you have to just basically deal with it. How does it happen? This is a nice image. What is this of? It's a neuron, right? And as you can see here, this neuron has got, what is this part called? The axon, right? And what is this part called? More known as cyton, right? Cell body as well, cyton as well. You can call it. Sorry for doing a biology class in the middle of the Gyan course. So you have these axon here, and the axon is actually myelinated. It's got a sheath of myelin, a myelin sheath at some areas. What are these gaps called? Well done, nodes of Ranvier. You guys have studied biology very well. I wish you had done chemistry as well, that strongly. All right, so you've got these nodes of Ranvier. So when you look at these nodes of Ranvier, that's where you find your neurofilaments here. And this is how a neurofilament normally looks like. If you isolated a neurofilament, it's about 60 nanometers in length, and it's about 10 nanometers in diameter. It's more like a cylinder, right? This neurofilament, as it degrades, the neuronal degradation gives you different biomarkers, NFH, NFM, NFL, and alpha int as well. Now look at the differences there. The biggest fragment is the one which is NFH, which is 200 kilodaltons, NFM, which is 150 kilodaltons, and then you have the alpha, which is 66, and our NFL is 68 kilodaltons, right? That's the unit, that's more kind of a unit that's normally used for biological macromolecules there as well, kilodaltons, right? So it's quite a big kind of a cylindrical kind of a structure, and I think I've got a representation here as well. This green ribbons that you see here are basically your NFL here. So you can see here, you've got these different 1, 2, 3, 4 units here. That's how it's, and as I said, it's quite a long, if it's a biomarker, it's quite a big biomarker there. It's got a very odd shape. Normally you would expect to have a globular protein or something which is more spherical. This one is more in the form of a cylinder there, right? And if you look at the image, this is an image which is basically showing you the green ribbons that you see here. That's your basically your NFL, which is your 68 kilodaltons molecule here. So I'm just going to run you past this. This is basically a model of your polyphenol red and your polypyridine, and this is basically your NFL. This one is your NFL, this whole thing. This is your NFL, and this thing that sits here, which looks like a chair, a G quadruplet structure, this is, what is this? Where do you find G quadruplet structures? Which bioreceptors show those kind of structures? Aptamers. Aptamers can take that particular form, right? Remember, aptamers can also give you those kind of hairpin structures. Why do they do that? Because you can have you seen these kind of structures? Complementary hydrogen bonding between the strands, and then you can have your molecule, target molecule, which can be easily trapped between the hairpin like structures. And then when you open it up, when you heat it up, it opens up, and then the target can be released as well. Okay, so this is just to show you some indications, because I know some of you are engineers and you wouldn't be able to appreciate what the size is. Just to remember, we are looking here, our target here, or our biomarker here is something which is quite big, which is about, as I said, did we say 68? Yes, 68 kilodaltons here. So this is 68 kilodaltons of NFL. That's how it's neurofilament light fragment. Okay. So apta sensors for NFL, right? So in Japan, they have managed to do apta sensors. They have managed to identify, they work a lot. So our collaborators in Japan, they work on different bioreceptors, they work on enzymes, they work on antibody fragments, and they also work on aptamers as well, right? So if you go and do a Google search on Tokyo University of Agriculture and Technology, TUAT Japan, and if you look for Professor Ikebukura or Professor Asano or Professor Sugawa, they all work on different bioreceptors, and they're all part of this UK-Japan project that we have on developing early diagnostic tools and prognostic tools for Alzheimer's. And these guys have actually come up with this particular aptamer here, which is basically, this is just a model I've used, creating a pi mole, so that you can help you visualize it, right? So this particular aptamer can bind to these red sites of the neurofilament of the NFL protein, right? This, as I said, this whole thing is your NFL, and you have these specific binds here, these sites here to which your aptamer can easily bind, right? So this is again data which is, as I said, unpublished data. So you should consider yourselves very privileged, because I'm sharing some unpublished data. Normally doesn't happen there, and Professor Khalid and Dr. Prasun will vouch for it that nobody presents unpublished data, but I wanted to give you the best. All right. So what is the nice thing about this G quadruplex aptamer here? The nice thing about this one, this was developed in, as I mentioned, TUAT Japan. We even got the names of the students who were involved in this. I think somebody who made this slide, very enthusiastic, I think at the moment we have Mio and Chinming who are in University of Liverpool who are visiting us for some joint experiments. Damai has just left. So they have actually done these are the people who have been working on this particular work. They've done the G quadruplex aptamer, which has been developed, and they are now trying to make an electrochemical sensor out of it, which we will call as the apta sensor. And they managed to do that. They're using 4 working electrodes, again using the same chips that you've seen. We call them the Elara chips, which says SSRG on top of that, the Smart Sensing Research Group 1. And they've managed to get a dynamic range. I think let's ignore this one. Let's have a look at this dynamic range here. Or even if you look at this dynamic range here, 64.28 picograms per milliliter to 30.3050 picograms per milliliter. Everybody happy with that? No? Why? No, that's nanomoles. Picograms per milliliter, if you multiply the milliliter with 1000 and then you multiply the picograms, that becomes nanomoles, right? Everybody happy with this dynamic range? What are we looking for? Yeah, and ideally, so what should be the dynamic range of your sensor? So what should be the when I say range, give me the values, please. Zero to 60. Somebody said 30. Well, 30 is where it just starts, so you need something a little bit more wider. Glucose goes up to maybe 12, 15, but we normally have sensors which give you a dynamic range of 0 to 40, 0 to 20. Yes, well done. 0 to 100, I will take that. I mean, I'm not the one working in the lab, so I always give, okay, fine, go and make 0 to 100, that's fine. So, and I'll probably invite you to do this sensor to do 0 to 100 picograms per milliliter. Yeah, does everybody agree that the dynamic range should be? Yes, please. Don't stick it into his mouth. Let him speak. He can speak loudly. Come on. Don't be shy. Come on. I mean, look, if you answer, then it encourages me. That means that you're understanding what these lectures are all about.
So
that's what
less than 11.5 means 10 to something more.
10 to 15.
10 to 100 or more.
Okay. Again, I'll take you back to the same. You remember I spoke about early diagnosis and prognosis. You need to not only just be able to diagnose things earlier, you also need to be able to follow up what is happening to the biomarker levels in response to a particular treatment. So you need a bit more wider range. That's why what the range of 0 to 100 picograms per milliliter is the right range, right? We'll have a special award for you. Well done. Okay. Now what's the problem here? What's the problem here? 64.28 to 3050 picograms per milliliter. What's the problem here? Are we getting the desired dynamic range? What did we decide? 0 to 100 picograms per milliliter, right? And we know the median value for a healthy volunteer is 11 picograms per milliliter. Don't go into so much details. When it comes to picograms, 0.1, 0.2, 0.3 would hardly make a difference. You know, if it's very hard, we can talk about different things, but if you speak to Rishita here, she will tell you making solutions for 0.2, 0.3 picograms per milliliter solution, it's not an easy thing. Yeah, does everybody agree? 11 picograms per milliliter is the median value. This is the dynamic range, and this one here, is it covering that range? No, it can be used probably as a prognostic tool. It can be used as a tool to kind of tell you what's happening, how is the AD person responding to a therapy, but it will not tell you, it will not give you early diagnosis. And what we want is early diagnosis. Let's start doing early diagnosis. And as I said, there are reports which suggest that if you can get in Alzheimer's disease, that the onset of the symptoms starts 15 years before the actual onset of the before the disease starts, before you start seeing the cognitive impairments. Right? So you want to have ultra sensitive detection there as well. The problem is when you go for ultra-sensitive detection, if you increase the dynamic range, you lose the sensitivity. That's another problem that you have, right? Okay, so does this warrant having another bioreceptor here? Yeah, all right, well done. So while these people in Japan, while our colleagues, our collaborators in Japan were working on, one second, yeah. While our colleagues in Japan were working on the aptamers at their end, we were working on the molecular imprinted polymers, which was basically, which is neurofilament. And here, as you can see from this value here, we are looking at something. This is the kind of results we are getting, and this one is basically in PBS. Yeah, pay attention. We do in our in vitro optimization in PBS and in, what's the other sample matrix? So if you were to do design, if you were to test your sensors, we're not doing skin, plasma, right? So this is some results in the PBS, and this is some results in the human plasma here. All right, you see this, and you see this. Okay, what I'm going to do is I'm going to show you when you combine these two together. This is what it looks like. So you have the green one is the one in human plasma, and the blue one is in PBS. Which one looks better? Why does PBS look better? Because the linearity range is all the way till here, right? Actually more in this way, something like this, right? And here the linearity range is probably till here. Maybe you can extrapolate it till here. Are we happy with this? Should we be happy with our MIPS? I mean 0 to 120 picograms per milliliter. Should we be happy with our MIPS? Yes, you will take it? I wouldn't. Why? Look at this range here. I don't care how far it goes here. What is my linearity range? And that's very close. What is my desired linearity range? 0 to 100 picograms per milliliter. That's my desired. Okay, I won't get all the way till 100. It's going to be very hard. I know that. But what can I do to get the best results here? And this is all about optimization. And then the best answers you get is when you test them with real clinical samples. So we have a opportunity. So just to explain you might ask me the question, why Alzheimer's? Why are you doing a project on Alzheimer's with a country like Japan? Anybody has got the answer for that? Yes. And aging population you have everywhere. Exactly. The demographic changes, and Japan, they are known for longevity as well. There's a region called Okinawa. Anybody heard about Okinawa? What's the speciality about Okinawa? People live over the age of 100 years, right? As I said, we are all victims of our own success. We've made fantastic medicines and everything, but we still need to address this main core issue is quality of life. Living longer is not the thing. Living a good quality of life is what we need, right? This is when we look at our grandparents and our parents. You can see with age, you can see that things slow down. How do you maintain a good quality of life? How do you maintain to be mobile? That's the main task. So if you have an aging population and if you have a very low birth rate, then you have very, it basically complicates the whole situation, right? This is a problem not just in Japan. This is a problem everywhere. In Japan, for some reason, apparently if you look at the statistics, 1/3 of the female population over the age of 65, 1/3 of the female population over the age of 65 suffers from Alzheimer's or dementia. And dementia is basically 70 to 80% cases of dementia are caused by Alzheimer's. You can have other reasons as well. You can have Parkinson's. These are different things. I'm not going to go into the clinical details of those kind of things. You have Parkinson's. You have different types of neurodegenerative disorders. But I think 70 to 80% of the dementia is mainly because of Alzheimer's. Therefore, if you find something which can predict the Alzheimer's, and now we have medication for Alzheimer's as well. So you need to bring in the treatments early on so that you cannot, I don't think you can cure it, because this is a natural degradation process. This is like diabetes. You cannot cure it, but you can help in managing the situation so that the person can maintain a good quality of life, right? If you go to Japan, if on the tubes you'll see people sitting down and doing sudokus, right? Those quiz like things. That's just to keep yourselves mentally aware, just to have a good cognitive skills, basically. Keep on practicing with your brain, right? That's how it works. Okay, now what I've done here is I've put the two, this is Japan and this is UK, right? This is the work done in Japan, this is the work that's done in UK. Okay, now what I want you to do is to look at the two plots, the two graphs. This is basically APTA sensors in PBS and APTA sensors in human plasma. This is APTA sensors in PBS and APTA sensors in human plasma. I want you to tell me one good thing with this and one bad thing with this. And please feel free to use the performance metrics that were discussed yesterday in the lecture. What's the good thing here? Okay, you're not getting the desired range. One, that's a bad thing. Yeah, we've already discussed that. I don't want anybody to repeat that. What's the good thing that you see here?
It will be more specific.
Well done. Well done. Did you know the answer before you came for this lecture series?
No.
Are you sure?
No.
It looks like you're trained, because this is I was not expecting this answer. Very well done. She deserves a special mention. Fantastic. Very well done.
Thank you.
Absolutely right. You can see from the two plots that your aptamer, which is your bioreceptor here, is able to pick up the target molecule in a similar fashion both in PBS, which doesn't have any other interferent, and in plasma, right? That shows how selective and how specific it is. Do you see the same thing here? No, there's a difference there. That means that our MIP system is not as selective as the aptamers are. Right? I was hoping that this discussion would go on for another 10 minutes, but thank God you saved me a lot of time there. Right? And this was discussed yesterday in the performance metrics as well. Well done on that. Well done all of you. Actually, if you've got that thought process and it's come to your mind that this is what it is, very, very well done here. So now, doesn't it build a nice case? What if we combine these two together? Because we would like to have the specificity, the selectivity of the APTA sensor and the dynamic range of the nano MIP device here. Again, I keep on mentioning nano MIP. Why are we calling it nano MIP? Louder. Lead by example. It's called a nano MIP because what is it made up of? What are the MIPs assembled on? Go on, louder. Don't be shy to give answers. Come on. Exactly, on highly porous gold. So that highly porous gold is your nanomaterial system, right? That microvilli example that I gave you, which will stick to you for the rest of your life. Every time you think about nanomaterials, you will think about the nanomaterial system you have in your own body. Our human body is fantastic, right? You can learn a lot from the human body in terms of, how the science works, how things work in the body, and then you can replicate that. I mean, Professor Khalid used to work for a group which was called as bio-inspired technologies. Technologies inspired from the human body, right? So microvilli, highly porous gold, right? That's your, you can look at the analogy there. All right. So again, if you put these two things together, if you put an APTA sensor and a nanomip system together, you can then get what we call as a much better. Will that happen? Has that happened? Well, from the initial results we've got from our PBS, and I believe these results. Can anybody tell me why I believe these results? Error bars, because I can see error bars here. When I see error bars, I know that this has been actually been done, not been made up there, right? And you can see here, what has happened here, look here. So this is the APTA MIP. So what we've done is we've done two things here. We've combined the aptamer plus the nanomip, right? Which is basically what you do is you take your metal surface, you have your nanoparticle system, your highly porous gold. On that, you immobilize your you immobilize your NFL on top of this. Sorry, it's mainly in the form of rods, right? So let's say these rods, and then you have your, sorry, apologies for rushing. Nobody's asked me where is the aptamer. So you have your surface, your highly porous gold surface, highly porous on top of that, and then you have your aptamer, which is bound to the NFL, right? So you have your aptamer, which I'm representing like this, which is basically like a chair, right, on which I'm sitting, G quadruplex structure, and then on that you have your NFL, right, and then you have your polyphenol and everything, and then you eat away these parts to create cavities which look like this. So that's your, this is your nanosystem, that's your aptamer, and that's your template, and that's your MIP, right? That's the combination there. What we also did was, we did one system where you have the aptamer with the nano MIP, and then we also created some surface, also part of the surface also has got some, so you can see here you got, it's not AMTA, I think it's got something wrong. Apta MIP is basically your aptamer with the nano MIP, and then you also have a aptamer nano MIP, and we also mix this with additional nano MIPs there as well. Why? Because when we just have the apta MIPs, this is the apta MIPs, you can see here it goes all the way till here, you're still getting a good dynamic range till here about 30, but if you want to increase this dynamic range all the way up here, which is up till about 50, right, you need to have more design, you need to design a better system here as well, right. So if you have something which is between 0 to 50 picograms per milliliter for a start, that's a good system. As I said, this is some ongoing work there, right. I'm showing this to you so that just to kind of motivate you, to encourage you to make a case that you really can make a lot with these kind of combined, by combining the bioreceptors here. If you look at this, how many systems do we have here? We have an aptamer, we have a nanosystem, we have a MIP, and then in another one, so you have got 3 different types of systems combined here, out of which you have a dual bioreceptor, which is your aptamer and the MIP, and you have a nanoparticle system here, and here you have a apta nano MIP, so you already have 3 parts here, and then you have a nano MIP here as well, which is 2 parts here. So this is like a 5 component system. So one is a 3 component system, so this one is a 3 component system, and this one is a 5 component system, and you can see you're getting much better results. So just to give you an example as to how you can play with the different components and optimize your systems to get ultra sensitive detection. And the idea here, as I said, is to widen the dynamic range so that you can cover a range. You can cover a range which allows not only early diagnosis but also helps you in prognosis and in being able to predict what's happening, or in being able to actually screen the particular patients and tell what's actually happening there. Is there early signs of diagnosis, early signs of AD there, or and how is the person responding to a particular chemical intervention of chemotherapy or a therapeutic intervention there. Okay, I think that's probably my last slide, just last few slides now. So I've given you 3 case studies there. So you have certain advantages. You can amplify the signal. You also can do multi-analyte capability as well. You can have different types of aptamers. You can do that. You have a much improved biocompatibility, better reproducibility, and you can model this engineering potential as well. Challenges to consider: fabrication complexity. I mean, I'm showing you the good results. I'm sure my students have got good days and bad days, like all of you have, right? Some days they get good results. So trying to standardize the whole fabrication is something that's really, really important. But that only happens once you've optimized everything and you know that this is my best possible scenario, or this is my best possible architecture. Interfacial stability could be a bit of a problem there, and then data interpretation, especially using machine learning and integrated systems. I already mentioned this to you, that you can have a very high value and the patient might turn out to be a non-AD patient, right? So you need to bring in machine learning and all those things, all those tools which correlate all the different factors that are involved, which include age and sex as well, because the prevalence is much more higher in female population, right? So a 65 plus female population with a value of 60 might be a short case of AD, while in males might be a different case. Things like that we need to be taken into account. And then cost of material. I mean, you're adding on the material, right? Because you're having multiple layers here, so you're adding on the material, so cost can go slightly higher. In terms of future directions, we are going to be looking at smart dual receptors which are responsive to pH or redox state. We're going to integrate this to wearable platforms. I think we're going to be working soon on insulin based wearable devices. And then we're going to use One Health Diagnostics so that we can use them in animals, environment and environmental samples as well. And then the dual receptor, we need to look at the machine learning tools for this dual receptor ones as well. And that's the summary. I've already mentioned that, just to summarize, your dual receptor biosensors can overcome performance limits of single systems. You can have, I've already shown you the case studies there, and you have new directions towards intelligent, adaptable biosensing platforms. Finally, I would like to acknowledge my research team in Liverpool and also from Japan. The funding bodies for the funding, MRC and AMED. MRC is in the UK, which is the Medical Research Council, and AMED is the equivalent of the Japanese research funding body there. To all my collaborators and my clinical partners. I think that's probably my last slide, and I'm going to open the floor for any questions that you may have. I know we have run massively out of time, but honestly speaking, we still have plenty of time for questions. I'm here just to encourage you, just to give you some information, and I would really encourage any questions that you may have.
Yep. Any questions?
Yeah.
So in the last slide which you have shown, where the dual receptors sensitivity has increased.
Yes.
So next slide, yeah. So this one on this green dots, I hope so that is a green one or red one, but green dots, there the error bar is less. So what might be the reason as compared to the red one?
I think the problem is one is in PBS.
Okay.
And the other one is in blood.
Okay.
The PBS one seem to have ions which might be interfering there, while with blood, because it might, and those ions might be masked in blood. I think that's the only reason I can think about. I mean, as I said, these are fresh from the press. These are these are latest results that I had. I think maybe done a couple of days back. So I haven't had a chance to discuss the whole results in details, and I'm not that much worried because, okay, both of them are actually in plasma. Both of them are in plasma. For some reason, basically the error bars just indicate that one system is more stable than the other one. Sorry?
Okay.
Okay, I will rephrase. Sorry, ladies, I'll hold on for 1 second. Both the studies have been done in PBS, not in plasma. Sorry, just a correction here. These studies are from PBS. We'll be able to see much better results when it comes to plasma. And the girls are right. It's actually, you have multiple systems when you're moving from 3 to 5 five component systems. So something here is contributing more to the noise. It could be adsorption of ions on the aptamers. It could be anything. We just have to look into details. I haven't looked into that. I just wanted to present this to show that we are getting better performance once we combine the different components together.
Okay. One more question.
Yeah, please.
So when you have shown that this sort of the architecture where the MIPS and these aptamers are combined,
yeah,
right? So now while removing the polymer, you make sure that both the MIPS part and the aptamer are combining to provide more sensitivity or specificity to the sample.
Yeah.
Now in this case, standalone aptamers, is the MIPS is facilitating the aptamer performance more, or if you use only aptamer, then aptamer orientation or its interaction would be reduced, something like that?
So from the initial studies that we've done, MIPS is giving you the desired dynamic range, getting you closer to the dynamic range. So it's giving you the dynamic range there. Aptamers is giving you the desired selectivity.
Okay.
So you are exploiting the properties of both of these together to get a highly selective and a highly sensitive biosensing system. Okay. With a wide dynamic range.
Okay. Okay. Thank you.
You're welcome.
Any more questions? Yeah, so just wanted to ask something very basic. What is the sensing modality? Is it pulse voltammetry?
DPV.
DPV.
Differential pulse voltammetry.
So
I didn't go into details because we already have discussed so much about DPV and things like that.
Yeah.
And I've already mentioned that I think when we were going through, I mean, we've not changed any, in terms of electrochemical sensing methods, we prefer using differential pulse voltammetry or square wave voltammetry.
Yeah. Okay. So why is it represented like as signal loss? What, just because the...
So that is the same thing what as I said. You have your surface, you have your binding sites. As you keep on binding,
so the sites get...
the sites get clogged, get blocked. So you have a drop in the signal.
Right.
because you're increasing the impedance of the system,
Okay.
of the layers,
Okay. Okay.
right? So the electron transport is getting much more, the electron transport of electrons is getting much more.
So higher the concentration, more sites will be
Exactly.
used up.
The higher the concentration, the more signal loss you will have. And the nice thing here is you're not using an external probe.
Okay.
The polymer itself is redox active.
Yeah. Okay. So just one more question. Amexine, is it biocompatible, or have you looked into that?
Again, for me, if somebody says amexines are biocompatible, I mean, I've seen little reports where they say they are biocompatible. It's like osmium polymers,
Okay.
osmium tetroxide, they're biocompatible. I'm not sure.
Right.
How can a transition metal become biocompatible overnight? So the ones that we used was vanadium, although we
Vanadium.
are not using it for wearable sensors. So that's why we moved away from wearable, because we wanted to use, as I said to most, as you know, I've been repeating this. You can easily transfer your protocols for point of care devices, biosensing devices, to wearable devices, because at the end of the day you're doing biosensing in both cases, right? So we wanted to use something which is more biocompatible, and highly porous gold is highly biocompatible as well. I mean,
People
gold,
use it.
silver, I mean gold is used, gold nanoparticles are used in anti-aging formulations, silver nanoparticles are used as antibacterials, so we know that they are.
Okay. Thank you. Any more questions? Okay, thank you very much for the very in-depth and informative session, and you have also shared some.
No, I'm
Data
actually
which
very
is not
glad.
published, yeah.
Very good answers, you know, from the students. Well done. Thank you all as well.
Thank you.
As I say, you know, when you sit and you answer the questions, it encourages me to do even better.
Yes, yeah, absolutely.
Although I'm not in the best of my health today, but still I didn't realize that.
So we'll reconvene again at 10:30.
Yes,
Okay.
10:30.
So